So we've seen that the end product of an anabolic operon can serve as a co-repressor to shut the operon off when that product is at high levels. And let's look at that now, schematically speaking. We have a TRP repressor gene, just like we had the LAC-I gene, which encoded the lactose repressor. Here we have the gene for the TRP repressor, which makes TRP repressor protein at some constant level in the cells. And if no tryptophan is present, or very low concentrations of tryptophan are present in the cell, most of the TRP repressors will not have bound tryptophan on them, and therefore they will adopt a conformation which does not have high affinity for operator DNA. And therefore the RNA polymerase bound to the promoter of the operon can transcribe the operon, producing the enzymes that are required for biosynthesis of the tryptophan amino acid. That makes perfect sense to us here. But if tryptophan is present, that the cell has plenty of tryptophan around, we want to turn the operon off, and that occurs because at high concentrations in the cell, most of the molecules, if not all the molecules of repressor, will most of the time have bound tryptophan to them. And tryptophan acts as a co-repressor, rendering the repressor protein in a conformation that has high affinity for operator DNA, blocking transcription of the operon by RNA polymerase, which is bound to the promoter. And in that case, with repressor and co-repressor bound to operator DNA, the operon will not be transcribed, and the gene products of the operon, the enzymes involved in biosynthesis of tryptophan, will not be produced, which is energetically favorable under these circumstances when tryptophan is at a high level in the cell. Prior to moving on to regulation of genes in eukaryotes, let's look at one additional level of regulation of the tryptophan operon, an anabolic operon, considering the elegance of the mechanisms we've already studied. But in this case, adding an additional level of complexity that points out to us the importance of coupled transcription translation in prokaryotes. So in prokaryotes, as you know, we have mRNA transcription and the immediate commencement of translation of that messenger RNA by ribosomes, because there's no nuclear envelope separating genes that are being transcribed from the ribosomes that are going to translate the messenger RNA so produced. Now, the TRP operon, actually, if we go back and look at our schematic of the operon, and I'm going to point out one additional feature, and that is that in addition to the structural genes that encode enzymes that are involved in the biosynthesis of tryptophan, there's one additional gene here that produces a leader peptide that doesn't actually encode enzyme. It's just a regulatory peptide. And so we'll call that gene, if you will, L for leader. And it encodes a leader peptide. So let's look at that now. So here is the 5' end of the tryptophan operon messenger RNA transcribed from DNA. And if we look at the conformation of this RNA, this is just the beginning of the RNA, at near the 5' end of the RNA. Excuse me. There is this RNA will adopt a secondary structure involving two stems and loops. Here's a stem loop, and here's another stem loop. And under these circumstances, when region 1 will base pair with region 2 and 3 with 4, there is a uracil-rich stretch, which we call the attenuator, that will under this conformation, cause RNA polymerase to fall off the DNA and stop transcription of the gene. Now, how does coupled transcription and translation attenuate transcription of this operon? Well, let's consider the case where tryptophan levels are very low in the cell. In the L gene, that is the gene that encodes a leader peptide, there are two adjacent tryptophan codons. That is, two codons that specify the insertion of tryptophan into this peptide, this protein. And those trip codons will be occupied by charged transfer RNAs bearing tryptophan only when tryptophan levels are high. If tryptophan levels are low in the cell, and remember, if tryptophan levels are low in the cell wants to express the operon so as to manufacture the enzymes that will synthesize more tryptophan. But when tryptophan level is low in the cell, there will be a low concentration of charged tryptophan tRNAs. And therefore, when the ribosome encounters these two tryptophan codons adjacent to each other, the ribosome will stall at this region when tryptophan levels are low, as I've already said. And when the ribosome stalls here, we will have the inability of stem loop 1-2 forming, because the ribosome will be sitting on, if you will, be occupying the parking space of region 1 here. In that case, region 2 base pairs with its second choice, which is region 3. It is capable of hydrogen bonding with region 3, forming a different stem loop. And when that happens, the region 3 cannot form a stem with region 4. And the attenuator, U-rich attenuator, is in such a conformation that RNA polymerase does not fall off the DNA template and continues transcription of the operon, thereby producing the products of the operon to manufacture more tryptophan. So when tryptophan levels are low, there is no attenuation of transcription of the operon, and that is because the ribosome stalls at these two adjacent trip codons due to the lack of charged tryptophan-bearing transfer RNAs. So now, what happens when, in this bottom case here, when tryptophan levels are high? When tryptophan levels are high, there's plenty of charged transfer RNAs bearing tryptophan amino acids. And the ribosome does not stall at this region, but continues on. And when it continues on, that means that it continues at a very rapid pace, that region 2 here will be occupied by the ribosome, and 2 will not be available to form a stem with region 3. And therefore, region 3 can form the attenuator stem with region 4, and exposing the U-rich attenuator, which causes RNA to fall off the DNA template, and transcription is terminated. So this is yet another elegant mechanism for regulating this anabolic operon. And this is attenuation that involves the coupled transcription translation of this operon. Again, emphasizing that when tryptophan levels are high, the ribosome will not pause, will not be stalled at these two tryptophan requiring codons, and will basically allow the attenuator stem and U-rich attenuator structure to form, which drives RNA polymerase off the DNA template, stopping transcription. So that is attenuation of trip operon transcription. And now we're ready to leave prokaryotic operon regulation, gene regulation in prokaryotes, and move to gene regulation in eukaryotes. Unlike prokaryotes, you'll remember that the eukaryotic genes, the protein coding genes in eukaryotes, produce monocistronic messages. In other words, there are not multiple protein coding genes found on the same messenger RNA. Each gene has its own transcript and therefore its own promoter. So operons don't exist as we know them in bacteria. They don't exist in eukaryotes. And shown here is the promoter region. This would be the start site of transcription for the thymidine kinase gene. And this is the thymidine kinase promoter for mammals. And essential elements of the promoter include this TATA box here, TATA sequence, that is highly conserved in most eukaryotic promoters at about minus 25 base pairs upstream of the start site. And it is to this TATA box that a specific transcription factor binds called TF2D, or TATA binding protein, known as transcription factor 2D as well. TATA binding protein, or TF2D. I'll write that down. So we have TF2D binding, also known as TATA binding protein, which binds to this TATA box. This is called a TATA box. Boxes refer to regions of DNA, conserved regions of DNA. But this transcription factor doesn't transcribe the gene. We need RNA polymerase for that. So we need our friend RNA polymerase, and it's RNA Pol polymerase 2, you will remember, for most protein coding genes in eukaryotes. There are 3 RNA polymerases, and RNA Pol 2 transcribes genes that are protein coding genes for the most part in eukaryotes. In addition, in this promoter, there are other sequences, a CAT box, a GC-rich, 2 GC-rich regions that bind other specific transcription factors that help aid binding of a set of transcription factors at the TATA box. But the first event for a transcription of a eukaryotic gene, the first event in the assemblage of a transcription initiation complex, involves a TATA binding protein here binding to the TATA box. And as we will see, TF2D is the first of a group of transcription factors that we will call general transcription factors, or I also call them basal transcription factors. And we'll say transcription factors are abbreviated TFs. So TF2D is a general or basal transcription factor, and there are other basal transcription factors that are required to be assembled at this core promoter, beginning with TF2D, that are required for loading RNA polymerase so that RNA polymerase can begin transcription of the gene at the transcription start site, 25 base pairs downstream, 25 base pairs 3', in other words, from the TATA box. So let's look at that now in a little more detail. Here we have TF2D, or a TATA binding protein, bound to the TATA box, and there are other basal transcription factors, A, B, F, E, and H, that associate with these, other basal transcription factors are recruited by TF2D once TF2D is bound to the TATA box. And there are TATA-associated factors, other proteins, quite complex, that bind to this basal transcription factor or general transcription factor complex. And that aids in loading RNA polymerase at the promoter. RNA Pol 2 will not be capable of transcribing a gene without these general transcription factors first being loaded at the TATA box, initiated by TF2 binding at the TATA box. So we have the loading then of RNA polymerase, which will transcribe the gene in response to the initial binding of TF2D and then the subsequent binding of other basal transcription factors and TATA-associated factors at this core promoter region. Now, what regulates the binding of, or the assembly of basal transcription factors at the core promoter, and therefore the loading of RNA polymerase at the core promoter to allow transcription of a eukaryotic gene, are activator proteins that bind to DNA sequences known as enhancers. We've talked about this a little bit earlier, and here we're now bringing this into play in terms of regulation of transcription of eukaryotic genes. These enhancer sequences that regulate a transcription of a particular gene can often be quite distant from the transcribed region of a gene. They can be thousands, and in some cases hundreds of thousands of base pairs away from the core promoter. And these activator proteins, we will call them auxiliary transcription factors or luxury transcription factors, bind to enhancers and through large folds of DNA are brought into juxtaposition of the core promoter and help load the basal transcription factors at the TATA box of the core promoter. So the activation of transcription of a eukaryotic gene is often positive in nature as opposed to negative regulation of bacterial operons. The only case we saw of positive transcriptional regulation of bacterial operons was the CAP binding protein. Otherwise, we were dealing with repressor proteins that turned an operon on or off. Here, for the most part, the regulation of transcription of eukaryotic genes, not exclusively, but largely is positive in nature. When positive activator transcription factors or auxiliary transcription factors bind to enhancer DNA, they load the basal transcription factors and assemble the transcription initiation complex. So that another view of this is as follows. Here we have enhancers bound by activator proteins that also bind to coactivator proteins that interact with these basal transcription factors. It's rather complex, and it's kind of like the leg bone is connected to the hip bone, connected to the backbone kind of thing, whereby this is a cascade of effects. Positive activator proteins bind to enhancers, interact with coactivator proteins that interact with the basal transcription factors and assemble them at the core promoter, and that allows RNA polymerase to be bound and begin transcription of the eukaryotic gene. So we have activators, coactivators, and general transcription factors that we need to consider when we talk about regulation of gene expression in eukaryotes. So you might then consider the situation in which when would a eukaryote wish to turn on a particular gene? Well, many genes are tissue specific. They're only expressed in a particular cell type. We're going to talk more about this when we talk about developmental biology. But in general terms, you understand that, for example, a lung cell would not need to be expressing genes for pancreatic functions, but would be needing to express genes that are required for lung cell function. And so lung cells must find a way then to turn on, to express positive activator auxiliary transcription factors that are specific for lung-specific genes, and in so doing can turn those genes on through the mechanisms that we've just considered. Whereas in another cell type, say liver, the liver cell would want to activate positive transcription factors that are bind to enhancers upstream of liver genes that are needed for liver function, so as to turn them on by mechanisms that we've already considered. Now, when we talk about transcription factor binding, especially these activator transcription factors that bind to enhancer sequences, we've got to go back to the structure of DNA and considering how these transcription factors bind. And you will remember, because the nitrogenous bases are attached not in their middle, so if we were to look at a case which does not obtain, but imagine that here we have a sugar phosphate backbone. We're looking down the axis of the helix. And here's an attachment of a nitrogenous base, let's say, and that is hydrogen bonded to another hydrogen base, to a nitrogenous base on the other strand of DNA. If the attachment of the bases to the sugar phosphate backbone, that is to the sugars through that glycosidic bond, were symmetrical in terms of the base, then we would have a groove on one side of the helix and a groove on the other side of the helix that were equal. There would not be a major and minor groove. They would be essentially equal, with equal amounts of the bases exposed in both grooves and equal distances between the sugar phosphate backbones in each groove. But the attachment of bases to the sugar phosphate backbone is not symmetrical. It is asymmetrical. So we have schematically this situation. There is an asymmetric attachment of these bases. And that asymmetric attachment generates a minor groove and a major groove, as shown in this diagram here. Here is our asymmetric attachment of our nitrogenous base to the sugar of the sugar phosphate backbone. Here's our glycosidic bond that links the nitrogen of a nitrogenous base to the first carbon of the ribose sugar. And that creates a situation where we have a major groove and a minor groove. And the major groove is much richer in terms of chemical information than the minor groove. And it is for this reason that many transcription factors preferentially recognize the base sequences in the major groove of DNA and bind to the major groove of DNA. And we can classify eukaryotic transcription factors, the auxiliary or activator transcription factors, primarily on their DNA binding domains. As we've already seen, if we look at these activator proteins, it's very clear that they contain a DNA binding domain that is responsible for recognizing enhancer DNA binding domain, and they contain other domains that interact with other proteins, namely coactivators in some cases and basal transcriptions in other cases. So these domains are protein interaction domains. And we classify eukaryotic auxiliary or activator transcription factors based on what type of structures exist in their DNA binding domains. We use their DNA binding domains to classify them because there are families of transcription factors which share common structural elements in their DNA binding domains. So let's look at briefly some of the DNA binding domains that are found in some major classes of eukaryotic transcription factors. Here is our minor and major groove situation. It's showing that the major groove has a longer distance between the sugar phosphate backbone than the minor groove and a richer stereochemical environment which can provide recognition sites for proteins that come in and bind in the major groove. One class of transcription factors in eukaryotes belong to the helix-turn-helix family of transcription factors because their DNA binding domain contains a helix, a turn, and another helix, which is and that recognition helix inserts into the major groove of DNA, recognizing a sequence of nitrogenous bases in the major groove and causing the transcription factor to bind to that particular sequence. If this were an enhancer sequence, the transcription factor would be bound there. Now, we're only showing a small portion of these proteins. We're only showing a small portion even of the DNA binding domain. We're focusing in on just the part of the protein that binds to DNA. Of course, there would be a much larger protein consisting of the rest of the DNA binding domain and the other domains that might exist on the protein. And often transcription factors will dimerize, so there will be helix-turn-helix transcription factors will act in concert, one monomer binding one turn of helix away from the other. If each base pair is 3.4 angstroms apart, then one turn would be 3.4 nanometers or 34 angstroms apart. And you often have transcription factors binding to DNA separated by one full turn of the helix. The homeodomain motif is like the helix-turn-helix motif, but there are actually three helices. Helix 1, turn, helix 2, and then helix, a turn, and then helix 3, which is the recognition helix of homeodomain proteins. And we'll come across the homeodomain again when we get into developmental biology. We'll see that this is a very important family of transcription factors that regulate the development of the anterior-posterior coordinates of embryos. Another family, I should say, of transcription factors are the zinc finger proteins. And they're called zinc fingers because there is a beta sheet, a two-stranded beta sheet, and an alpha helix, and another alpha helix, and the beta sheet and the alpha helix coordinate zinc atoms. The zinc atoms provide stability to the zinc finger motif, and they chelate or bond to that zinc atom with histidine or cysteine amino acids, and there are usually 4 that are involved in binding to zinc and therefore stabling the zinc finger. This beta sheet projects as like a finger, and the alpha helix then is responsible for inserting it in the major groove of DNA and promoting sequence-specific, enhancer-specific binding of zinc finger transcription factors to their recognition sites, enhancer sites in eukaryotic chromosomes. Another family is the leucine zipper family of eukaryotic transcription factors, and the leucine zipper family of transcription factors are required, in order to bind DNA effectively, are required to dimerize. And we call them zippers because there are alpha helices that associate with each other by the interaction of hydrophobic amino acids on one side of the alpha helix. You will recall basic alpha helix structure. If we were to look down the axis of an alpha helix, where we're looking at the backbone chain here, with the amino acid side chains stick out from alpha helices. And the alpha helices that are present in leucine zipper proteins that interact with each other are what we call amphipathic, meaning that there is a hydrophobic side to them and a hydrophilic side to them. So we will have a polar side to them in which polar amino acid side chains project, and a nonpolar or hydrophobic side from which nonpolar amino acids project. One of those being the amino acid leucine. And these amphipathic helices of leucine zipper transcription factors, the amphipathic parts of helices associate with each other, the hydrophobic parts of these amphipathic helices associate with each other because the hydrophobic amino acids wish to hide away from the aqueous polar environment. So here we have the polar sides of the alpha helices, and here we have the nonpolar sides of the alpha helices, and those nonpolar sides associate with each other. And we often find leucines that are hydrophobically bonded in this region. So that's why it's called a leucine zipper. It's like these are zipped up there by the hydrophobic forces that are acting to promote association of two monomers of a leucine zipper transcription factor. When these two monomers come together, and again, we're not showing the huge part of the proteins that are not immediately involved in DNA binding. When these two proteins associate, they form a scissors-like grip on the DNA, with one alpha helix bound to a major groove on one side and another alpha helix on the other side. They grip the enhancer DNA in that fashion. Now, before further consideration of regulation of gene expression, in eukaryotes, we've got to consider the packaging of DNA as a step at which transcriptional regulation can occur. And in the next lecture, we will pick up with this regulation of transcription in eukaryotes by differential packaging of DNA into either transcribable states of DNA packaging, of chromatin packaging, or in transcription unfavorable states, condensed chromatin, which will prohibit the transcription of genes. So we've got to look at DNA packaging, chromatin packaging as a point at which transcriptional regulation can be effected in eukaryotes.